I know it's overwhelming. By mid of this lecture, you will feel overwhelmed. بس please stay with me. اضرب حالك كفين صح شوي إن شاء الله you will enjoy it and understand it as well. So blood coagulation, what is blood clotting or coagulation? This is a definition that I modified myself. So basically it is a coordinated. So you would see how coordinated, how beautiful this is, this process. It's a biochemical process as well. We're talking about enzymes, we're talking about proteins, we're talking about protein-protein interaction, enzyme inhibition. So it's really a biochemical process by all means. It is initiated as a result of injury. Once it is initiated, you have in a small area in blood, you have this process going on surrounding the injury, whereby you have the formation of a clot. This clot, basically, it is conversion. It is basically the liquid form of blood would transform into a gel, and this gel becomes solid, preventing bleeding or resulting in what we know as hemostasis or cessation of blood loss. This is followed by repair of the injury site plus dissolution, removal of the clot. So it is really coordinated. Yesterday I injured myself as I was preparing this lecture, and I started thinking of all of these things that I'll talk about. Okay? And they have them here. We have the finger, by the way. Shahada. Okay. So these are the different steps, basically. We love to divide things into stages and phases. So basically, what we have here is vasoconstriction to stop bleeding. Makes sense. Followed by platelet aggregation to close the wound and to have clot formation going on. What's not shown in here is the final process, which is basically removal of the clot, dissolution of the clot. All right. So these are the different players. We have blood cells. We have the inflammatory cells. We have cell fragments, specifically platelets. We have proteins. We have enzymes. We have cofactors, enzyme cofactors. We have inhibitors. We have tissue glycoproteins. We have surface glycoproteins. Glycoproteins on the surface of endothelial cells and on the surface of platelets. We have lipids, phospholipids, and we have ions, calcium ions more specifically, and then we have the vasoactive compounds. So it all starts with platelets. What are platelets? These are cell fragments. They are produced from the multinucleated megakaryocytes. Now what happens here is that these megakaryocytes are pinched off into small rounded structures known as platelets. Platelets, they still have their own actin and the actin regulatory proteins. And they also have granules, like vesicles. And looking into these granules, there are different types of granules. As I've been preparing for this lecture, some talk about 4 different types of granules, others talk about 3. I'm going to stick with 3. So these granules are the dense, electron-dense granules, and they contain calcium ions, these ones here, calcium ions, ADP, ATP, serotonin, as well as other molecules. And we have the alpha granules. Alpha granules contain heparin antagonist, a growth factor known as platelet-derived growth factor. We have the fibrinogen, we have the von Willebrand factor, we have clotting factors that we'll talk about now, and we have the lysosomal granules or lysosomes that contain hydrolytic enzymes. Now what happens is that during activation of platelets, you have release of these, you have fusion of these granules to the plasma membrane and release of their contents. And then you have signaling that goes on. So the process looks something like this. It's again divided into different stages. We have adhesion to endothelial cells. and adhesion to matrix proteins as well. Followed by aggregation of platelets, so you have platelet-platelet interaction and clustering, and then we have coagulation, the enzymatic process that takes place at the surface or on the surface of platelets. So we'll talk about these signaling pathways. So what happens is that you have injury that takes place. This injury exposes collagen to blood cells, to platelets. Now platelets can interact with, collagen can interact with the surface receptors on the surface of platelets, integrins, different types of integrins that are glycoproteins as well. So they are glycosylated receptors. So collagen interacts with these receptors, activating platelets. In addition, and that results in release of some of these granules. Now, something else is that you have another protein known as von Willebrand factor that also exists in matrix. It's embedded in there. So it becomes exposed to platelets, and it can again also interact with integrins, activating these platelets. Now, what happens as well is that when these platelets are activated, you have modulation of the actin cytoskeleton and change of platelet structure from rounded structures into structures with filopodia. These are known as filopodia, extensions. Okay? These extensions contain more receptors, and they help in platelet-platelet aggregation. Now, the contents of granules, these are some examples of these molecules that get released. We have ADP, ATP, serotonin, a neurotransmitter but it also exists in there. Factor 5, the von Willebrand, so we have release of more von Willebrand, so that activates on the receptor itself, again, resulting in more and more activation of platelets, thromboxanes, and so on. So different components or different molecules are released. But yes, as a result of platelet interaction with the collagen and the von Willebrand. Right. So let's talk about we'll talk about the signaling pathway. You have release of some thrombin. Thrombin binds to its receptor right there. This receptor activates phospholipase C. Phospholipase C degrades PIP2, which is phosphatidylinositol 4,5-phosphate, into two molecules. You already know of these two molecules: diacylglycerol, DAG, and IP3 phosphatidylinositol 3. These are signaling molecules. IP3 results in release of calcium inside platelets. Calcium has a lot of effects, including it activates phospholipase A2. Phospholipase A2 acts on phospholipids in the plasma membrane, resulting in the removal of arachidonate. Arachidonate is converted to prostaglandins. Prostaglandins are converted to thromboxane A2 by thromboxane synthetase. So you have these reactions that take place. We already talked about these reactions in Biochem 1. So thromboxane, why is that important? Because it is a vasoconstrictor. That's important. We want to slow down blood flow, so we need thromboxane to be released. Now, thromboxane, the reaction, if you remember, is, or it can be targeted by aspirin. Aspirin inhibits cyclooxygenase 1, which is responsible for converting arachidonic acid to prostaglandins. So it inhibits the inflammatory process from going on. Okay? And blood clotting. It inhibits blood clotting. By the way, I read a few months ago that the idea that aspirin helps in reducing heart attacks is false. I don't know how true it is. Just keep that in mind. So thromboxane is important for this process, and it can be inhibited by aspirin. Now, we also have another vasoconstrictor, which is serotonin. It acts on different receptors, on endothelial cells, small vessel cells, whatever, platelets, and so on, resulting in vasoconstriction. Another molecule that is released is platelet-derived growth factor, which is an inducer. It's a growth factor that induces the proliferation of endothelial cells. So when these proliferate, the wound gets closed. Okay? So that's that. Now, there's something else that calcium does. In addition to the synthesis of thromboxane A2, it can act on another protein known as myosin light chain kinase. It activates it. It can then, it's a kinase, it phosphorylates myosin light chain. Myosin light chain can then, when it's phosphorylated, it can bind to actin, it modifies actin, so the shape of the platelets changes, and you can have platelet-platelet interaction as well. So it's more activation of platelets. Okay, so that's what calcium does. Now the other arm of what phospholipase C does is that it results in the formation of diacylglycerol. Diacylglycerol binds to protein kinase C, which phosphorylates one of these proteins inside platelets, and it induces the fusion of granules with the plasma membrane of platelets so that the contents are released. So you have some thrombin in plasma that binds to its receptor, activating this, so more thrombin will be released, as well as other components of granules. So that's the signaling pathway. We also have ADP that acts on its receptor right here, and it also results in in activating platelets, changing their structure, and resulting in fibrinogen-induced platelet aggregation as well. So you have activation of platelet interaction, or clustering, aggregation. And then the other process is the coagulation process, which is what? It's basically a series of reactions that take place on the surface of platelets that result in blood coagulation. We'll talk about the process, the biochemical process, right now. Quick. Yes? الابنفرين برضه موجود gets released. It binds to its receptor. في له receptor موجود على السيل surface. برضه
صح الابنفرين بكل مرات بيمشي قدام بغض النظر في انجري.
Yes, but it's all regulated. يعني any of these components involved in here, the ADP or the ATP or the thrombin, they exist in blood, yes. لكن they do not act by themselves. You need something to initiate the fire. بدك هاي الشعلة الصغيرة. هاي الشعلة الصغيرة بتيجي من interaction of the platelet with the collagen. This interaction right here. This is important. هذا معناته إنه there's injury. Some of the endothelial cells lining up the vessel are gone. And now platelets can interact with collagen, and that induces all of these reactions. Right. إيش عندنا كمان? So everything happens on the surface of platelets. Right. So let's talk about blood coagulation now. The biochemical process itself. ناخذ نفس. وهيك عدل عدتك تتحرك شوي عشان الحكي خطير يعني كثير overwhelming معلش. So we have different molecules that are involved in blood coagulation, and different players as well. فعندنا the organizing surface, that is the platelets themselves. Okay, so everything happens on the surface of platelets. We have different molecules. We have zymogens. Remember zymogens? Zymogens are inactive enzymes, and they can be activated by proteolytic cleavage of part of it. So they get modified. Now, so they exhibit digestive zymogens. They're always there. Like once we eat, we have activation of these zymogens. So we have these zymogens in blood. And they are in blood, and they are produced by liver cells. But they are zymogens, inactive, and they're waiting for the inducer. Again, شعلة. Now there are different types of zymogens. We have different enzymes and factors. We have, they're known as factor 12 and 11 and 9 and 10. We have cofactors as well. We have factors 8, 5, and 3. Different types of cofactors that they're not enzymes. They associate with these factors, factor 10 and 11 and so on, activating them. Okay? We also have, when these factors are activated, they are giving the designation a subscript of A. So X factor 10 is inactive. That's a zymogen. If I say factor 10A, it means that this is the activated form of the enzyme. But yes? But we have also anticoagulants. We'll talk about them at the end. Protein C and S. So keep that in mind. And we have calcium ions. We have thrombin, which is also one of those zymogens as well. Yeah, I could have written it in here. And we have fibrinogen, the protein that forms the clot itself. Now, when they first studied the pathways of blood coagulation, they initially thought that we have two pathways that are independent of each other. And they're known as the intrinsic pathway and the extrinsic pathway, depending on what the source of injury is. So the intrinsic pathway, you have an injury that takes place inside the body. It just happens. If there is an external effect, like trauma, injury, like what I did with my finger here, this is external, the extrinsic pathway that is activated. But it turns out that there is a bridge. There's a bridge that links these two pathways together. We'll talk about it. As well as, they converge, they meet at a point of factor 10. So this factor 10 is commonly activated by the intrinsic pathway and the extrinsic pathway. Factor 10 activates prothrombin to thrombin. Okay? So they're not really independent pathways, rather no, they are linked to each other. That's how it looks like. This figure is taken from Marx. And you can, by the way, go back to Marx to read the section on blood coagulation. It's really beautiful. But it's intensive. Every sentence is loaded with information. So I'm trying to simplify it. So I relied a lot on Marx to prepare this lecture. So basically, these are the two pathways. In the intrinsic pathway, we have factor 11 that gets activated. It activates factor 9, which activates factor 10. And in the extrinsic pathway, we have trauma. You have activation of factor 7, which is an enzyme. And it activates factor 10 as well. So you have activated factor 10. So they meet here. Now factor 10 activates prothrombin to thrombin, and thrombin causes blood clotting, the formation of the clot. Notice that in here, you have these cofactors right here. You have factor 8, that is important for the activation of factor 10. And you have number 7, not only that it is an enzyme, but it also bridges the extrinsic pathway with the intrinsic pathway. It can activate factor 9 so that it can activate factor 10. So here, so the cross link is right here. And the activation of factor 10 in the extrinsic pathway by factor 7 requires a protein known as tissue factor. Right? You should memorize this, by the way. What's missing in here, actually, in this figure, is that there is another factor known as factor 12. And factor 12 acts upstream. It's actually right here. It activates factor 11. Okay? So it's right here. We'll talk about it. منيح. questions؟ نعم. ارجع شوي.
ايش شفت السهم اللي تقطعه؟
ايش هو؟
السهم الاخير.
السهم اللي بالاخير اي سهم هذا؟ هذا؟
ليش عامل كونكشن مع
ما هذا بدي ارجع لك. هذا الحلو بالموضوع. استنى علي شوي. اوكي. هلا. The purpose of this is just to show you the different domains that exist in all of these factors. And there are common domains. Like, notice that the catalytic domain, which is in red, exists at the C terminus of all of these enzymes and proteins. That's the active domain. But what I want to focus on is this here in green, right here. It's basically a stretch of, it's a sequence of amino acids that is rich with glutamate. Okay? That's in green. So basically that's how it looks like. Okay, so this is in green is a sequence of amino acids and it is rich with glutamate. That's in green. Why is that important? Because this domain gets carboxylated. What happens is that that's glutamate, it gets carboxylated, so you have the addition of another carboxyl group. Now glutamate, glutamic acid, it has a carboxyl group. The carboxylation of these proteins or this domain results in the formation of a carboxyglutamate. The importance of carboxyglutamate is that it can be chelated. شو يعني chelated? It means that you have two groups that can interact with a common ion, like the calcium ions. Okay? So you have this calcium ion that makes an interaction with two carboxylic groups. Why is that important? Because now this positively charged calcium ion can make an interaction with the negatively charged phospholipids on the surface of platelets. So all of these proteins that have the glutamate domain can associate with the surface of platelets. So now you have all of these reactions taking place on the surface of platelets. So it's contained. It doesn't happen anywhere in blood. No, it happens only at the site of injury. You have all of these reactions going on at the surface of platelets. The carboxylation of glutamate takes place in the ER and Golgi during the production of these proteins, which are produced from hepatocytes. So they are released again as cytogens, and they are released as modified with carboxylated glutamate. The carboxylation of the glutamate residues, and there are about 10 or more of these glutamates, requires vitamin K. K means coagulation because it's written in K in German. So vitamin K is important for coagulation because it facilitates the action of carboxylase, the carboxylation of glutamate. And vitamin K must be regenerated so that it can act on other glutamate residues. It can carboxylate other glutamate residues. And this is important because it can be targeted by an anticoagulant. Okay, right? So you have a number of proteins that can be carboxylated. And these are the ones. Prothrombin, factor 7, an enzyme, factor 9, 10. And we have these anticoagulants, protein C and protein S. Great. Inshallah. So, the end result of activating the intrinsic pathway or the extrinsic pathway end up with the activation of factor 10, and factor 10 can activate thrombin. And this happens at the surface of platelets again. طيب, شو بيصير عندنا هون actually? That's how factor 10 acts. It acts on prothrombin, converting it to thrombin. What do we need? We need calcium ions so that we can chelate the carboxyglutamate and associate all of these proteins with at the surface of platelets. That's one. We need phospholipids, so calcium can interact with the phospholipids. That's one. and we need factor 5 as well. That's a cofactor, it is not an enzyme. So that's how it looks like. You have factor 5 associating with the surface of platelets. It associates with prothrombin as well as factor 10. Getting them close to each other. Now that they are close to each other, يعني جمعين راسين حلال، شو بنسوي؟ What factor 10 does is that it cleaves prothrombin, again it's a cyanogen, it cleaves it, and prothrombin is released and in the active form. Okay, so this is the process. That's how we get them together, and that's how we activate prothrombin. أي سؤال؟ كويس. هلا. So it's really complex. لأنه activating of the extrinsic pathway or the intrinsic pathway occurs at the surface or on the surface of the platelets. So you have this, the association of factor 5 helping the activation of factor 10 via factor 7, that's an enzyme, and factor 10 associate with factor 5, activating prothrombin, PP, so prothrombin is now active. At the same time, that's in the extrinsic pathway. In the intrinsic pathway, we have factor 8, it associates with the cell surface, it helps in the, you have activation of factor 9 by factor 10. Now factor 9 associates with factor 8. It helps in the activation of factor 10. Factor 10 binds with factor 5, just like in here, activating prothrombin to thrombin. Yeah, basically the idea is that it takes place at the surface of platelets. Now there's something here that happens as well. Let's zoom on this. So the idea here is that you have prothrombin, or you have thrombin that is active. The thrombin, by the way, you would read it in books as factor 2. It's known as factor 2. Anyhow, so what the thrombin does, that's the activated thrombin, is that it releases factor 8 from the von Willebrand. Now you have von Willebrand in the plasma, swimming, bound to factor 8. Why is that important? Because without the von Willebrand factor, factor 8 has a short half-life. It gets degraded really fast. Okay? So what von Willebrand does, in addition to being present in the matrix and activating platelets, it binds to factor 8, it prolongs its half-life. So people with deficiency of von Willebrand have limited amount of factor 8, and as a result, there is limited activation of the coagulation process, and they suffer from bleeding. ماشي؟ طيب. Thrombin can also function on other factors as well. Let's just focus on tissue factor for now, ونرجع على بقية processes. So you have tissue factor is involved in the extrinsic pathway, but what it does is that it associates with factor 7, activating factor 10, but it's also linked to the activation of factor 11, or the activation of factor 9, not 11, factor 9, which activates factor 10. So that's the link between the two. Okay. Now, going back to vitamin K, I said that vitamin K must be reduced in order to activate carboxylase enzyme so that we have carboxylation of glutamine. But vitamin K must be regenerated by another enzyme, by a reductase. Now, this reductase can be targeted by warfarin. So if there is warfarin in the system, you have the inhibition of this regeneration of vitamin K, so you don't have this, and you don't have coagulation going on. So warfarin is an anticoagulant that is given to patients, especially before surgeries. Okay, so it's a target of this, of regeneration of vitamin K. But yes? ناخذ نفس. There's, very quickly, there's an important system that we should talk about, like on the side, and it's known as the kallikrein-kinin system. Kallikrein is an enzyme. And it is also produced as a zymogen, as a pre-kallikrein. Kinin is produced from a peptide known as kininogen. So it's a peptide, not an enzyme. So you have kininogen that gets cleaved by kallikrein. And this happens on the surface of endothelial cells. Enter when there is an injury. Yep. So you have kallikrein is produced. What kallikrein does from pre-kallikrein, what kallikrein does is that it activates factor 12. This is the one that I told you about that is not present in here. Factor 12, it's up here. Okay? So factor 12 activates factor 11. And what happens is the following. So you have activation of factor 12. Factor 12 activates factor 11. Factor 11 activates the intrinsic pathway. منيح؟ كويس. هلا notice that factor 12 also activates more pre-kallikrein into kallikrein. So it's a cycle. There's amplification that is going on. Something important about kallikrein is that it can act on kininogen, producing bradykinin. It's a peptide, a short peptide that can act on endothelial cells, inducing vasodilation. ليش؟ لأنه we want inflammatory cells to come at the injury site as well. So we need vasodilation as well. طيب. So as I said, it's a coordinated process. It's balanced. Something else about this is that the bradykinin or the kallikrein-kinin system is that it's also important for the angiotensin-renin system produced by the renal system as well. That is important for hypertension. But هذه معلومة on the side. طيب, what is fibrinogen? Now we have active thrombin. What does it do exactly? It acts on fibrinogen. Fibrinogen is a sort of like a matrix protein. Okay, it exists in blood, and that's how it looks like. That's the fibrinogen monomer. So you have two triple-stranded proteins that interact with each other by disulfide bonds. This is known as the fibrinogen monomer. The thrombin acts on this side right here, and these two, by the way, are linked by disulfide bond. And this part is cleaved by thrombin, activating the fibrinogen. So you have this reaction that takes place. Thrombin acts on this, releasing fibrinopeptides. That's not important, but you have the fibrin monomers. These fibrin monomers can polymerize. So this part of the fibrin monomer can interact with the globular heads of another fibrin monomer. So you have this cross-linking that takes place. And the formation of what you see in here. You have this fibrin monomers polymerized, forming a clot. And this clot is known as a soft clot. Why? Because this interaction, by the way, between this part and the globular heads is via electrostatic interactions, non-covalent interactions. Okay, so it's a soft clot. But how can we make it a hard clot? By another factor. This time, this factor is known as factor 13. It is not a serine protease like all of the other enzymes. كل الانزيمز اللي حكينا عنهم factor 13 and 11 and 12, factor 12, 11, 10, 9, 7, all of these are serine proteases, just like chymotrypsinogen or chymotrypsin and trypsin. These are all serine proteases. ليش؟ لأن عندهم serine in the active site, so they're known as serine proteases. طيب شو بيعمل factor 13? It is not a protease. It is a transglutaminase. It has a transglutaminase activity, meaning that it removes an amino group from glutamine and it links it to a lysine covalently. So you have a fibrin monomer and a fibrin monomer that are close to each other, linked by electrostatic interactions, comes transglutaminase, factor 13, and it links them together covalently. Now this is known as a hard clot. طيب مين بيعمل activation factor 13? Thrombin. So what thrombin does is that it accelerates the formation of the fibrin polymers, but it also activates the transglutaminase to form the hard clot. And that's the idea. ماشي. Another important point is that we have an amplification process. شو يعني؟ احنا حكينا we have an enzyme that activates an enzyme that activates an enzyme that activates an enzyme. So one enzyme can activate, let's say, 1,000 enzymes. Each one of these 1,000 enzymes can activate 1,000 enzymes. And again, you have activation, each one can activate 1,000 enzymes. So you have an amplification process because you have sequential activation of all of these factors. Now, something else that is important is the line that you saw, the dotted line that you asked about, is that thrombin, once it gets activated, not only that it can act on fibrinogen, not only that it can act on transglutaminase, it can go back in a process known as feedback activation. It can activate all of these factors. It can activate factor 11, it can activate factor 8, it can activate factor 5 as well. So you have amplification of the cascade itself, of all this process as well. Now,
Can you see positive feedback?
yes, exactly.
طب how does it stop?
حاضر استنى انا شويه حطخين حيطخوك يعني لا نكسر بس I have to talk about it. So we have anti-clotting factors. The process must stop. Yeah, it doesn't continue forever. It has to stop somewhere. So we have anti-clotting factors. And remember, these factors, so talked about protein S and protein C. Okay? What happens is that once you have thrombin in the system, it binds to a protein known as thrombomodulin. So it modulates thrombin. And this protein is present on the surface of endothelial cells. Once thrombin binds to thrombomodulin, it can convert and activate protein C. Protein C binds to protein S. Remember, these two have the same glutamate domain that can be carboxylated, so they can bind to the surface of cells as well. Now, once you have the complex of protein C and protein S, they can degrade their enzymes, and they can degrade all of these other factors like factor 5 and factor 8. So the amplification or the activation process stops altogether. There's no more activation of factor 10 anymore. Okay? So, يعني حاميها حراميها. You have activation of thrombin. Thrombin accelerates the system. Like eventually, what happens, it can also induce its own inhibitors. So now you have an inhibition of clotting. Something else is that you have a protein known as antithrombin. Antithrombin 3 binds to heparin. Heparin is a protein. We talked about heparin before, glycosaminoglycan, remember Biochem 1? Well, heparin is a protein that keeps on swimming in blood, and what it does is that it binds to an inhibitor of thrombin known as antithrombin. It activates it so that it makes it easier for thrombin to bind to the complex, and thrombin becomes inhibited. And it can also bind to other factors, like factor 10. So factor 10 gets inhibited by antithrombin as well. Now the interaction, again, is via the serine of the active site. Anyhow, so now we have activation of different proteins that inhibit coagulation. We also have a tissue factor inhibitor as well that binds to the cell surface, or that binds to tissue factor on the cell surface. طيب قربنا نخلص معلش صحصحوا شوي عدوا عندكم معلش. There's a nice section in Marx that talks about protein or clot dissolution. استحمل معي معلش اقرأوها. لكن we have to remove the clot. It has to dissolve somehow. And it gets dissolved by proteases. This time, the protease is known as plasmin. So we're going to start here with plasmin. Now, plasmin is activated from plasminogen. It's zymogen. Plasminogen is or has very high affinity to the clot. So it binds to the clot with high affinity. So when it gets activated within the clot. Once it gets activated, it can degrade the fibrin clot into fibers and the clot would be dissolved. حلو كتير. But plasmin itself must be regulated. And it's regulated by an activator, and it's also regulated by an inhibitor. So the activator is known as plasminogen activator, PA, plasminogen activator. And there are two types of plasminogen activators. We have tissue plasminogen activator, and we have urokinase plasminogen activator. Urokinase plasminogen activator is synthesized by many cells. The TPA is mainly produced by endothelial cells. So these are activators of plasmin from plasminogen. This process is also inhibited by plasminogen activator inhibitor as well. It functions on the plasminogen activator. So this is an inhibitor. طيب. So plasminogen activator produces more plasmin. Plasminogen activator inhibitor inhibits this. طيب. هذا by itself is also regulated by activated protein C. يعني عندنا inhibitor بيعمل inhibition للinhibitor. الانهبيتر وقتها لما يصير له inhibition بيعمل activation للاكتيفيتر. الاكتيفيتر بيعمل activation للplasmin. So it's different layers of activation that take place. So activated protein C that we talked about before inhibits the plasminogen activator inhibitor, and it also activates the plasminogen activator as well. معلش. Okay? We also have an antiplasmin that acts on plasmin and it inhibits it as well. So different layers of regulation. Final note is that we have a protein produced from bacteria known as streptokinase. So it is produced from streptococcus or streptococci bacteria. Now, what this protein does is that it can activate circulating plasminogen to plasmin in blood. So it helps in dissolving the fibrin clot. And it can be used, it can be given to patients. لما يكون مثلا I think you know لما واحد كبير بالعمر بده يعمل عملية وراح يضل قاعد هو بالفرشة لفترة طويلة، شو راح يصير عنده؟ Formation of clots, small clots would form here and there. فاحنا ايش بدنا anticoagulants. ممكن نعطي له warfarin، ممكن ينعطى الستريبتوكاينيز as well, layers to prevent the formation of clots. So بيقدر انه يدخل على عملية ويعمل عملية. لكن pay attention that the amount of streptokinase given to patients must be well controlled. And if you give too much, that causes a lot of bleeding. And this is dangerous as well. خلاص. يلا يعطيك العافية. محلاتكم ناخذ الغياب.